Thank you for inviting us. We appreciate the opportunity to share some of our work here today. So I'm Anne. I'm a professor at Fontys University of Applied Sciences, and I don't know if everybody is familiar with universities of applied sciences or hogescholen in the Netherlands and their research groups. So I want to give a short introduction into ours because we are really a multidisciplinary research group. We are called Applied Natural Sciences, and if you're interested, feel free to follow us on LinkedIn because there we share our work. So you can see we are a mixed group, and me and Joost, we are professors for the life sciences domain, and basically we're focusing on molecular diagnostic technologies for improved health. So this really covers that spectrum, so therefore I'm happy to share our work. Last year, only in the Netherlands, there were almost 500,000 consults for sexually transmitted infections. So this entails consults at the general practitioner, but also sexual health centers. And when left untreated, sexually transmitted infections can have serious consequences like infertility. So chlamydia is a sexually transmitted infection caused by bacteria called Chlamydia trachomatis. And this is the #1 STI in the Netherlands. And only last year there were over 64,000 positive tests. Chlamydia trachomatis is a special bacteria. I always say to my students, it's a special bacteria. In the past, they really thought it was a virus because it's intracellular and it is known to have two forms. I'm not going to in too much details, but you have the infectious elementary body. You can see on the top, does everybody see my pointer? Because it's an arrow. Can you see it? Yes. So you have the infectious elementary body. And then that really changes to the reticulate body, which is able to multiply in the cell. And then it's released again as elementary bodies to infect more cells. And this makes it impossible to culture this bacterium on the traditional agar plates, as everybody usually does in the bacteriology department. So you need tissue culture if you want to culture this bacterium. And therefore, basically, nucleic acid amplification techniques are the gold standard to diagnose chlamydia. There are conventional real-time PCRs you can perform, as you can see my colleague Juliette in this picture, but you can also use different commercial systems available for high throughput screening. So the development of the next generation PCR might improve the field because you can really think about accelerated detection from clinical samples. I also want to share that we work on wastewater screening, and also we perform some of this work for wastewater, but then we didn't work with Chlamydia trachomatis, but with the mouse strain variant Chlamydia muridarum. So why would you be interested in screening wastewater? That's basically because not everybody gets tested. That's what we also know from the corona pandemic. So therefore, STIs are also if you don't have symptoms or complaints, you will not go to the doctor, but you can easily walk around with chlamydia because most women at least are do not have symptoms. So we are actually also looking into this with next generation PCR besides digital PCR, which is more known to be used for wastewater. So, but due to the time we have in this small pitch, the aim of our study was to convert a conventional real-time PCR protocol to a next generation PCR assay to accelerate detection of Chlamydia trachomatis from wastewater and clinical samples. But for today, I will only focus on the clinical sample part. And in this picture, you can see our cycler room in which we have all our systems set out for the work we do. So the methods for this project is that we worked in close collaboration with Maastricht University Medical Center. They provided a pool from clinical samples. This is really a mix of clinical positive samples. We don't know what's in the samples, only we know that they are positive for Chlamydia trachomatis. We used Maastricht's protocol, the conventional real-time PCR, which targets both the OMP-A target and a plasmid target. and their current protocol takes 92 minutes. And then I'm only talking about the PCR program. I'm not talking about sampling, sample prep. That's excluded from this talk. So you have to keep in mind that every time I discuss time needed for a protocol, it only entails the PCR protocol. So for this project, we adjusted some parameters from the conventional real-time PCR method we received from Maastricht. So we shortened the OMP-A amplicon to 85 base pairs. We looked in time optimization of the different PCR steps, and we reduced the PCR cycles. So let's continue to the results. First, we were really enthusiastic about the first quick win, and it was just only changing from a Quant Studio, which you can see in this picture. So we have the Quant Studio 1, and we used the PCR pool of samples with the conventional protocol we got. It takes 92 minutes. We have a positive signal for the pool, and the no template control stays negative. And then just changing to the next generation PCR platform with a readout on the Fluorstar, we got this protocol finished within 64 minutes. Just changing machines. So that's a quick win you get. And then we continued shortening time for each PCR step and also the number of cycles. So we'll start in this picture. You see the initial denaturation step. It takes 10 minutes in the conventional protocol. We just reduced it to 1 minute, and we still got some nice results. So this was a quick win, 9 minutes reduced by just changing the initial denaturation step with good results. Then you have the denaturation time in the PCR cycles. It takes normally 15 seconds, but we were able to reduce it to 5 seconds. The conventional PCR program is a 2-step protocol, so annealing and elongation time are combined in the conventional real-time PCR. So we also reduced that time, and we saw that we got acceptable results at 40 seconds instead of 1 minute. Then reducing the PCR cycles, you can see that on the right side lower bottom. The conventional program entails 42 cycles, and we found acceptable results at 38 cycles. So by just shortening PCR steps and cycles and not changing temperatures or 2 steps to 3 steps protocol, just easy work, we were able to get an assay on the Next Gen PCR system, readout Fluorstar of 32 minutes. So also this is already a reduction of 1 hour from the conventional program. Then we tried another step, the Corona pandemic. We also had a SARS PCR program, not of MBS, but we had our own. But MBS had a program with 2 phases. So first phase was just a low amount of cycles, longer time at each temperature, and then ultra fast cycling for a large amount of cycles. So we tried to template this for our Chlamydia trachomatis protocol, and we were able to further reduce number of PCR cycles to 35 instead of the 45 that was depicted here. And by optimizing the PCR program to a 2-phase phase PCR program, we were able to get a next generation PCR assay of 17 minutes. So of course, further optimization can be done. Of course, also clinical samples need to be tested. You have to also work on sample prep time reduction to have a really efficient clinical test. But we were really enthusiastic that we were able to get a next generation PCR assay of 17 minutes instead of 92 minutes on the Quant Studio. So the conclusion of our work is that we developed an assay on the next generation PCR machine for Chlamydia trachomatis, not by changing a lot of things, and reduced the time to results from 92 minutes to 17 minutes. And this can have major impact if you think about sexual health centers where patients come in and you want to test them fast, and they can leave with maybe antibiotic therapy in one session instead of that they leave from your health center, and then you have to call them back to get their antibiotic therapy. Of course, I did not do this work on my own. I want to thank the whole life sciences team of our partnership, because all my colleagues are needed to do this work. But we want to thank especially Anouk Poppelaars, because she was the graduation student who performed this work in our group. Of course, I want to also thank our collaborators, and I have to say that this work was funded by the Fontes Center of Expertise, High-Tech Systems and Materials, and also Isogen Life Science. Thank you for your attention, and I'm happy to take your questions. I will stop sharing my screen, and I can see you guys.
Thank you very much for the presentation. We have a question in the chat. So I don't know if you can read, but I can read.
Yeah, I can open the chat now.
Okay.
And compared now, endpoint PCR is would endpoint be sufficient, and why? I think if you have your, it's a different way of looking at samples, but if you do a real-time PCR machine, I think nobody is looking at the screen permanently to watch a sample get positive or not. So if you have an endpoint PCR, but you have a really fast endpoint PCR, and I even thought that MBS is working on a real-time PCR system now, so then that would even be more efficient if you have a real-time measurement instead of the Fluorstar measurement or gel electrophoresis, because then you all the time you win in the PCR program, you lose by doing the gel electrophoresis. But if there's a real-time PCR next-gen system, and you can have like in 15 minutes a readout, endpoint doesn't matter if you have your controls in your assays. Of course, that's basic. You need your controls in your assays, otherwise you don't know where you're looking at. I hope that's an answer to your question. Otherwise, feel free to. Okay, thanks. Yeah.
Yeah, thanks. Okay, perfect.